Welcome, curious minds, to the deep dive. Today we're embarking on a mission, really, to decode one of the most fundamental systems in human biology. We're talking about the male reproductive system. And this deep dive, it's specifically crafted for you. Maybe you're navigating a dense college textbook, or you're a medical student looking for that, shortcut to being well informed. Our goal is always to cut through the jargon, get straight to those aha moments. For this deep dive, our main source is a real cornerstone, Medical Physiology, the updated edition by Boron and Boulpaep. It's incredibly comprehensive, really detailed. We're focusing on Chapter 54, trying to pull out the absolute essence for you.
Exactly. And our mission for you, the listener, is to take all that intricate physiology from Chapter 54 and make it, well, not just clear, but actually engaging and clinically relevant, too. We'll start big picture,
Yeah.
build every concept from the ground up. Then we'll break down the step-by-step reasoning, how these systems really function. And even without pictures, we'll try to paint a mental image so you can visualize the anatomy, the cells, everything.
Okay, let's unpack the foundational architecture then, the basics of the male reproductive system. When we talk about its core, we're really looking at two essential components. First, the gonads. In males, that means the testes. Now, the real insight here isn't just that they have a dual role, but how elegantly these two things, making sperm and making hormones, are balanced. And these hormones, they don't just drive reproduction, they shape the whole male physiology, even influence behavior, right?
Oh, absolutely. And the structure of the testes is fascinating, how it achieves both. Imagine like a super efficient factory. It's largely filled with these incredibly tiny coiled tubes, the seminiferous tubules. Think of them as miniature plumbing systems, lined by the specialized epithelium anchored securely. It creates this unique protected space for sperm development. And then nestled in the spaces between those tubes, you find the interstitial Leydig cells. They're the hormone powerhouses. Then beyond the testes, you've got the sex accessories. It's this whole network. Glands, conduits, like the epididymides, vas deferens, seminal vesicles, ejaculatory ducts, the prostate, bulbourethral glands, Cowper's glands, the urethra, the penis. Their job. Pretty straightforward but vital. Store and transport the spermatozoa out, make sure they can reach and fertilize the female gamete.
Right. And a quick but really crucial point here. For these testicular factories to work properly, they need to descend into the scrotum during fetal life. If that doesn't happen right, you get a condition called cryptorchidism. The testes stay up in the abdomen, and that can cause, well, significant damage to the seminiferous tubules. Function goes way down.
Yeah, it really highlights how critical temperature regulation is for making sperm. Just a few degrees makes a massive difference.
Okay, so with that foundation set, the male body then goes through this huge transformation,
Yeah.
puberty. It's this really remarkable transition, juvenile to adult. You see secondary sexual characteristics emerge, that big adolescent growth spurt, and crucially, the ability to procreate. And the timing can vary quite a bit, right? Onset between 9 and 14 years, and it takes maybe 2 to 4.5 years to complete.
That's right. And the very first sign you can usually see, what clinicians often call Tanner stage 2, is the testes getting bigger, enlarging to more than, say, 2.5 centimeters. That's mostly the seminiferous tubules growing, though the Leydig cells chip in too. And when we track this, clinicians use the Tanner method. It describes genital development and pubic hair growth separately. So you could have a boy who's like genital stage 3, but still pubic hair stage 2. They don't always perfectly align.
Interesting. And what about sperm production starting?
Ah, yes. A key milestone is spermarchy. That's the first appearance of spermatozoa in early morning urine. It happens around 13.4 years on average, and it lines up with specific Tanner stages. Then you get the pubertal spurt, that big increase in body size. Boys grow, on average, about 28 centimeters during this time. Part of that is growth hormone, but it's hugely influenced by rising testosterone levels. And you see the result in adult anatomy. Men end up with roughly 150% of the lean and skeletal body mass of women, on average, and about twice the number of muscle cells.
Wow. And the absolute driving force behind all those secondary characteristics, the deeper voice, the hair patterns, that's the androgens, right? The male sex steroids.
Exactly. Testosterone is the main player here. It causes the maturation of the external genitalia, for instance,
Yeah.
and that voice drop you mentioned. That's androgens making the larynx bigger and thickening the vocal cords. For boys, the vocal cords can increase in length by like 50%.
50%, that's huge.
It is. And you also see those classic male hair patterns, face, armpits, pubic region. Even things like temporal recession or male pattern balding later on are androgen-driven.
And they have muscle building effects too.
Yes, significant anabolic effects. They stimulate linear growth, help the body retain nitrogen, which is key for building protein, and promote muscular development. It's useful to differentiate, though. Androgenic effects are on the reproductive tract and secondary characteristics. Anabolic effects are the growth-promoting ones on other body tissues like muscle and bone. But here's the key insight. They're independent actions of the same type of steroid. The molecule is the same, but the target organ's response determines if it's an androgenic or anabolic effect.
Okay, that distinction is helpful. Now, this is where things get really intricate, isn't it? The control system. The hypothalamic pituitary gonadal axis, the HPG axis, you described it like a control panel, a feedback loop governing both sperm production and those androgens we just talked about. How does this master system actually work?
Right, it's a really elegant cascade. It starts up in the brain, in the hypothalamus. The hypothalamus secretes gonadotropin-releasing hormone, or GnRH. It's a decaptoptide, just 10 amino acids, but powerful. And what's absolutely critical is how it's released. Pulsatile and pulses. It's secreted into this special blood vessel system, the hypothalamic pituitary portal system. Think of it like timed burst, not a steady stream. For LH, it's about 8 to 14 pulses every 24 hours. That rhythm is the secret sauce.
Pulsatile release?
Yeah.
Okay.
And that GnRH travels just a short distance to the anterior pituitary gland. There, it stimulates specific cells called gonadotrophs. It's like a key fitting into a lock. And it tells these cells, okay, releases the gonadotropins, which are luteinizing hormone, LH, and follicle-stimulating hormone, FSH. This binding kicks off a process inside the cell involving calcium release, leading to the synthesis and secretion of LH and FSH.
And that pulsatile nature has real clinical implications, doesn't it? You said pulses stimulate release, but what if you give GnRH continuously?
Exactly. It's kind of counterintuitive. If you give GnRH analogs continuously, nonstop, it actually suppresses LH and FSH release. It desensitizes the pituitary.
So you can shut down the system.
Precisely. And that's used therapeutically. In conditions like prostate cancer, where you want to lower testosterone, giving a continuous GnRH analog effectively achieves a chemical castration.
Wow. Okay, so LH and FSH are released from the pituitary. Where do they go? What do they do in the testes?
They have distinct targets. LH primarily acts on the Leydig cells, those interstitial cells we mentioned earlier. It binds to receptors on their surface and basically tells them, make testosterone, which stimulates the synthesis of testosterone from cholesterol. There's a key rate-limiting step in that process.
The conversion of cholesterol to pregnenolone?
That's the one, involving the enzyme P450cc. LH ramps this up by increasing the enzyme's affinity for cholesterol and boosting its synthesis. The result, more testosterone.
Okay, LH targets Leydig cells for testosterone. What about FSH?
FSH targets the Sertoli cells. Remember, the nurse cells inside the seminiferous tubules supporting the sperm.
Got it.
So FSH binds to receptors on Sertoli cells, and this triggers protein synthesis. They start producing several important things. First, androgen-binding protein, ABP. This gets secreted into the tubules. Its job is crucial. Keep local testosterone levels super high, right where sperm are developing.
Makes sense. Keeps the testosterone concentrated.
Exactly. Second, they make P450 aromatase. This enzyme converts some of the testosterone that diffuses over from the Leydig cells into estradiol, right there in the Sertoli cell. Third, they produce various growth factors. These directly support the sperm cells, help spermatogenesis along, indirectly increasing the number of germ cells. And finally, they produce inhibins. These are glycoproteins, and they act as negative feedback, mainly telling the anterior pituitary to ease up on FSH secretion.
So there's feedback within the system.
Absolutely. And what's really critical is the crosstalk between the Leydig and Sertoli cells. It's not just one-way signals. Leydig cells make testosterone, which acts on Sertoli cells. But then Sertoli cells convert some of that testosterone to estradiol, which can act back on the Leydig cells. Even those growth factors from Sertoli cells can increase the number of LH receptors on Leydig cells, affecting how much testosterone they can make. It's all interconnected.
Okay, so bringing it together. For optimal sperm production, you really need both Leydig and Sertoli cells working together. And you need both LH and FSH from the pituitary, plus enough testosterone locally. They're all interdependent.
You nailed it. And you see this clinically, like in Sertoli cell-only syndrome. These men have normal male characteristics because their Leydig cells are making testosterone fine, but they have azoospermia.
No sperm.
Because the germ cells aren't there or the Sertoli cells aren't supporting them properly, often because the germ cells themselves are absent. And interestingly, these men often have high FSH levels. Why? Probably because there's less inhibin being made by the Sertoli cells to provide that negative feedback.
That really underscores inhibin's role in the feedback loop. Okay, let's zoom in on the star hormone itself. Testosterone. How is it actually built? How does it get around? What does it do?
Right. So synthesis happens in the Leydig cells, starting from cholesterol. There's a preferred pathway, a series of about 5 enzyme steps. And that conversion of cholesterol to pregnenolone by P450cc is the main control point, regulated by LH. But testosterone isn't the only active form. It can be converted into dihydrotestosterone, or DHT. This mainly happens outside the testes, in target tissues, using an enzyme called 5-alpha reductase.
Yep.
And get this, DHT is way more potent than testosterone in some cells, like 30 to 50 times more potent.
Whoa.
Yeah.
Okay.
And testosterone can also be converted to estradiol by aromatase, like we saw in the Sertoli cells, but also elsewhere.
So how does it travel in the blood? Is it just floating free?
Mostly no. Only about 2% is actually free testosterone. About 45% is bound pretty tightly to a specific protein called sex hormone binding globulin, SHBG. And about 55% is loosely bound to albumin, another common blood protein. It's that tiny free fraction, that 2%, that can easily diffuse into target cells and do its job.
And once it gets inside the cell?
Well, either testosterone itself or DHT binds to a specific androgen receptor, which is usually inside the cell nucleus. This binding activates the receptor, which then acts as a transcription factor. It basically goes and turns specific genes on or off. This leads to the synthesis of new proteins that cause the cell to grow, differentiate, or change its function in some way. So a key point here is whether a tissue responds to androgens depends entirely on whether it has these androgen receptors. No receptor, no response.
That connects directly to something very relevant. Testosterone and the aging man. We often hear about testosterone levels declining with age. Is that a sharp drop like menopause in women?
No, it's generally more gradual. After maybe ages 25 to 70, there's often a slow decline in serum testosterone. And this correlates with things people associate with aging, maybe decreased bone formation, losing some muscle mass, changes in hair growth, lower appetite, maybe reduced libido.
And can replacing that testosterone help?
Often, yes. Testosterone replacement therapy can reverse many of those changes. It can help restore muscle and bone mass, sometimes correct anemia. It can significantly improve quality of life for many men.
Okay, let's shift gears now to the process itself making the sperm, from the very beginning to the final product.
Right, the whole journey is spermatogenesis. It happens inside those seminiferous tubules, and it's really three processes happening concurrently. First, you have mitosis. Think of the starting cells, spermatogonia near the basement membrane. They're diploid, 46 chromosomes. They just divide and divide, increasing their numbers, like a stem cell pool constantly renewing itself.
Okay, more cells.
Then some of these spermatogonia become primary spermatocytes. They duplicate their chromosomes, still 46, but now duplicated. And they enter meiosis. Meiosis happens, and they become secondary spermatocytes. Now they're haploid, but the chromosomes are still duplicated. Then, really quickly, they go through meiosis the second. And now you have spermatids. These are truly haploid, unduplicated chromosomes. So one primary spermatocyte ends up producing 4 spermatids, and interestingly, 2 will have an X chromosome, 2 will have a Y.
Determining the sex of the offspring later on.
Exactly. The third process is spermiogenesis. This is where the round spermatids transform into the shape we recognize as mature spermatozoa. They shed a lot of cytoplasm, develop that long tail, the flagellum. It's a major remodeling process.
How long does all of this take?
The whole thing, from spermatogonium to spermatozoon, about 74 days. And roughly 50 of those days are spent right there inside the seminiferous tubules. And a key thing, the rate of spermatogenesis is pretty constant. You can't really speed it up with hormones. A young man, say 20 years old, might produce around 6.5 million sperm per gram of testes tissue per day. That rate does tend to decline slowly with age, though.
And throughout this, those Sertoli cells are playing a big role, right? The nurse cells.
Absolutely vital. They provide nutrients, support, basically everything the developing sperm need. And they form those tight junctions between themselves. That creates the blood-testes barrier.
Like a security fence.
Kind of, yeah. Similar idea to the blood-brain barrier. It creates a very specific protected environment inside the tubule. It shields the developing germ cells, which have different surface proteins than the body's own cells, from the immune system. And it controls what gets in and out. Very important.
Okay, so after the tubules, where did the sperm go? They're not quite ready yet, are they?
Not quite. They move from the seminiferous tubules into this network called the reed at testes, then through the efferductuals and into the epididymis. The epididymis is this incredibly long coiled duct, like 4 to 5 meters long, folded up on the back of the testes.
4 to 5 meters. Wow.
Yeah, and when sperm first enter the epididymis, they're largely immotile. They can't really swim properly, and they can't fertilize an egg. It's during their transit through the epididymis, which takes maybe 12 to 26 days, that they undergo critical maturation. They gain progressive motility, the ability to swim forward, and the capacity to fertilize. If you take sperm from the head of the epididymis, they usually can't fertilize. But by the time they reach the tail or the vas deferens, they're generally capable.
So the epididymis is like a finishing school for sperm.
That's a great way to put it.
All right.
Then from the epididymis, they move into the vas deferens. This is a tube with strong muscular walls that helps propel them along, eventually leading to the ejaculatory duct.
And then we get contributions from the accessory glands.
Exactly. The seminal vesicles, the prostate, the bulbourethral glands, they produce the fluid part, the seminal plasma. And this plasma makes up like 90% of the total volume of semen.
What are typical semen values?
Normally, volume is maybe 2 to 6 milliliters. Sperm count should be at least 20 million per milliliter. And the pH is slightly alkaline, usually 7 to 8.
And what's in that seminal plasma?
The seminal vesicles are the biggest contributors, maybe 70% of the volume. They provide fructose, which is the main energy source for the sperm. They also contribute citric acid and other things. And that fructose is clinically relevant.
How so?
Well, if you have a man with a low sperm count, oligospermia, and also a very low ejaculate volume, testing for fructose is important. If fructose is absent, it strongly suggests there's an obstruction or maybe even congenital absence of the seminal vesicles or ejaculatory ducts.
Ah, okay. Diagnostic clue.
Definitely.
Yeah.
The prostate gland contributes too, adding factors that help prevent sperm from clumping together, and enzymes that help liquefy the semen shortly after ejaculation, allowing the sperm to swim freely.
And blockages can happen elsewhere too, right? Like the vas deferens.
Yes, that's ductal obstruction. It can be congenital. For example, men with cystic fibrosis often have congenital absence of the vas deferens, or it can be acquired maybe from infection, scarring, or very commonly from a vasectomy. In these cases, sperm production in the testes might be perfectly normal, but the sperm just can't get out. That leads to azoospermia, no sperm in the ejaculate. It's a common cause of male infertility.
Right. Okay, let's wrap up this deep dive by looking at the neurophysiology, the male sex act itself. It sounds like a complex coordination.
It really is. A true neurophysiological symphony, you could say. The whole general system gets input from both the sympathetic and parasympathetic divisions of the autonomic nervous system. Plus, there's somatic innervation, primarily from the pudendal nerve. These different nerve pathways originated from different levels of the spinal cord and coordinated via various plexuses and ganglia have to work together just right.
Let's start with erection. How does that work?
Erection is primarily under parasympathetic control. What happens is the smooth muscles within the erectile tissues, the corpora cavernosa, relax. This relaxation opens up the arteries, allowing a huge increase in blood flow into these spaces. They fill with blood, pressure increases, and that causes the penis to become erect and rigid.
And what causes that smooth muscle relaxation?
The key player is nitric oxide, NO. It's a gas, a signaling molecule. It's released from the parasympathetic nerve terminals down there. It diffuses into the smooth muscle cells and stimulates an enzyme that produces cyclic GMP, or cGMP. cGMP is the intracellular messenger that actually causes the muscle cells to relax, leading to vasodilation and increased blood flow.
Ah, and that connects directly to erectile dysfunction treatments, right? Like Viagra.
Exactly. Drugs like sildenafil, Viagra, vardenafil, tadalafil, they are PDE5 inhibitors. PDE5 is the enzyme that normally breaks down cGMP. So by inhibiting PDE5, these drugs allow cGMP levels to stay higher for longer when NO is released. This enhances the smooth muscle relaxation and vasodilation. But, and this is important, they only work with sexual arousal. You still need that initial NO release triggered by the parasympathetic nerves. They don't cause erections on their own.
Good point. Is there any sympathetic role in erection?
There is, actually. Tonic, ongoing sympathetic activity normally helps keep the penis flaccid by keeping those smooth muscles contracted. So part of achieving erection involves a decrease in that sympathetic tone, alongside the increase in parasympathetic activity. And the somatic nerves, the pudendal nerve, they control striated muscles like the ischiocavernosus and bulbospongiosus at the base of the penis. Contraction of these muscles helps increase the pressure and rigidity, especially during the final phases in ejaculation.
Okay, so that's erection. What about emission and ejaculation?
Right. Emission comes first. This is the movement of the seminal fluid sperm from the vastus deferens, plus fluids from the seminal vesicles and prostate into the prostatic urethra, the part of the urethra running through the prostate. Emission is primarily under sympathetic control. It involves coordinated peristaltic contractions of the smooth muscle in the walls of the vas deferens, seminal vesicles, and prostate. And crucially, at the same time, the sympathetic nerves cause the internal sphincter muscle at the base of the bladder to constrict tightly.
Why is that important?
To prevent retrograde ejaculation. You don't want the semen going backwards into the bladder, you want it moving forward into the urethra.
Which can happen sometimes, retrograde ejaculation.
Yes, it can. If the nerves controlling that sphincter are damaged, maybe from diabetes, multiple sclerosis, certain surgeries, or if someone's taking certain medications that interfere with sympathetic function, the sphincter might not close properly. Then, during emission, semen can flow back into the bladder. Sometimes, sympathomimetic drugs, which mimic sympathetic activity, can help treat this. Got it. So emission loads the urethra, then comes ejaculation.
Exactly. Ejaculation is the forceful expulsion of that semen from the urethra out of the body. It's primarily a spinal cord reflex, usually triggered when semen enters the bulbous urethra. This reflex causes rhythmic contractions of several striated muscles, the pelvic floor muscles, the ischiocavernosus, the bumbospongiosus. These contractions powerfully propel the semen outwards.
An orgasm, how does that fit in?
Orgasm is generally considered the culmination, the peak of sexual excitation. In males, it's the cognitive experience, the sensation that correlates with ejaculation. Its precise mechanisms involve complex interactions between the central and peripheral nervous systems. It brings together all those neural pathways we've discussed into that final unified experience. Wow. Okay, so what does this all mean for you, our listener, embarking on this journey? We've really covered a lot ground here, decoding the incredible complexity, but also the elegance of the male reproductive system. From the basic anatomy, that intricate HPG axis control, the hormones, spermatogenesis step by step, and finally the neurophysiology of the sex act. It's quite a system, isn't it?
It truly is. And what I hope you take away is how interconnected it all is. The anatomy supports the function. The hormones drive the processes. The nerves coordinate the actions. Understanding one piece really does help unlock the next. It builds this holistic picture. You've just taken a really significant deep dive into a foundational and frankly pretty dense chapter of physiology. So remember, you're part of the deep dive family. You're absolutely capable of mastering this material. Keep exploring, keep asking questions. That's how you build that deep understanding.
Absolutely. And now, maybe a final provocative thought to leave you with, building on everything we've unpacked today. Considering how precisely balanced and interconnected these hormonal pathways and neurological controls are, how might emerging technologies, maybe things like cutting-edge gene editing or really targeted neuromodulating therapies, potentially revolutionize how we approach conditions like male infertility or even prostate cancer in the coming decades? What could the next big breakthrough in male reproductive health look like, given everything we've learned about these intricate systems today? Something to think about.